Now let's discuss the different sorts of lab investigations that we carry out for different patients. Specimens may be examined grossly, for example, to detect adult worms in feces. Although microscopy is rapid, it is insensitive and requires considerable expertise. Specificity may also be a problem if commensal organisms may be mistaken for pathogens. Microscopy can also be used to define specimen quality, for example, identifying the salivary contamination of sputum by the presence of epithelial cells. Then we have special stains which can be used to help identify the organisms, such as Giamza staining is used for blood films and tissues, which is used to identify malaria and leishmania. Immunofluorescence can provide precise identification of a pathogen by using antibodies that are directed against a specific organism. Let's discuss how do we culture out organisms. First of all, antibiotic therapy administered pre-sampling can falsely render samples culture negative. Most human pathogens are fastidious, which means requiring media supplemented with nutrients to support the growth and increase their number to the detectable levels. Growth on solid media allow the organisms to be separated into individual colonies. A pure clonal population permits subsequent identification and susceptibility testing. Selective agents such as antibiotics or dyes may be used to suppress unwanted organisms in specimens with a normal flora. We have to basically have an appropriate atmosphere which has to be provided to the organisms, such as the fastidious anaerobes. They require an oxygen-free atmosphere. Most pathogenic bacteria are incubated at 37 degrees centigrade, but some fungi are incubated at 30 degrees centigrade. Now, how do you identify the different pathogens? Identification predicts the pathogenicity, such as the Vibrio cholera causes the severe watery diarrhea, whereas Shigella sonnei infection is usually mild. Identification of organisms should prompt the public health action. For example, the contact tracing of a patient found to have the meningococcal meningitis is very, very important. Bacterial identification depends on colonial morphology on agar, microscopic morphology, biochemical tests, and increasingly nucleic acid amplification tests and gene sequencing. This is especially important for organisms that are slow growing, for example, Mycobacterium tuberculosis, or impossible to grow, for example, Trophirema whipplei. Susceptibility testing aims to determine whether a treatment with a given antibiotic will be successful or not. A susceptible organism should respond to a standard dose of an antimicrobial. A moderately resistant strain should respond to a larger dose, whereas a resistant organism is likely to fail the therapy with the given antibiotic. Clinical response depends on the host factors, and in vitro tests only provide an approximate guide to the therapy. Several bodies, including the British Society of Antimicrobial Chemotherapy and the Clinical Laboratory Standards Institute, define the methods and standardize the conditions to ensure the testing is reproducible. Both are based on the measurement of the diameter of the zone of inhibition of confluent growth for the test organism that is caused by an antimicrobial incorporated into a paper disc. The minimum inhibitory concentration, which is the lowest dose that completely inhibits the growth, is a more objective method and enables the resistance levels to be related to the concentration of antibiotic that is achievable in the tissues. Susceptibility can be assessed rapidly by hybridization or sequence-based methods that detect the specific antibiotic resistance mutations. Now let's find out about the techniques used in the serology. An infection can be diagnosed by detecting the immune response to the pathogens. For example, by the detection of rising or falling antibody concentrations more than a week apart, or by the presence of a specific IgM or specific antigens. These techniques are used for organisms that are difficult to grow, such as viruses, for example, HIV or hepatitis B.